Hi everyone, and my name is Fan Qingyuan from Southern University of Science and Technology, and I'm happy to here to share with my research. And my research topic is about the DNA codec and the privacy communication. And my topic is the robust and private nanopore DNA readout via advanced codec design and de novo Basecallings. And as many colleagues, my first page is about the advantage and the durability of the DNA storage. But most of you have seen this many times, so I just jump it, as the DNA has a high informational density and the longer storage durability. And also, we found that the nanopore readout, the nanopore sequence made the real-time DNA readout feasible, because you found that compared to the next generation sequencing like the NGS, and the nanopore sequencing has the advantage of real-time that we just use the pore motor as the sensors, so we just get all the current from the pore itself at the real-time speed. And it's also portable, like the the minimum MK1D machines on the left upper of this GIF. It is quite portable, as small as mobile phone size. And lastly, the most important, that compared to the NGS, the nanopore sequencing has a longer read length, which means that we may need less assembly workload when after the sequencing that we need to assemble and get the data, retrieve the data from the DNAs. However, the nanopore-based DNA storage still has some challenges, at least on these pages. On the left, we found that compared to the next generation sequencing, the nanopore sequencing has a higher indel rate, which means insertion, deletion, and substitution rate. You will find that the nanopore sequencing may have an indel rate about 10 to 50%. However, compared to the NGS, may only have an indel rate of 1.1%. So that's quite different, which may break the conventional DNA storage codecs that are designed for the NGS data. And on the right figures, you can also find that when there are some chemical modifications, such as 5-hydromethylations or 5-HMC, 5-MC, or 6-MA, will break the sequencing current of the nanopore sequencing. While this may seem to be some weakness of the nanopore sequencing, we found that it may be also a promising for the security, because when it completely disrupts the signals, which may lead to readout failures of the conventional Basecallers, that may mean that we can develop some new Basecallers and pair with these modifications to do some biological stenography or informational concealment. Yes. So this comes to our computational solutions. And first, we present the composite hedges nanopore codec, which is a DNA storage codec for our robust DNA readout, which use the degenerated bases and the hedges codec system. And the second one is the DPSME for the private communications. In this research, we just developed a pair of the keys, ways to train a pair of the keys, that the Basecallers that are paired to the chemical modifications to correctly Basecall the modified DNAs. Let's come to the first part, the composite hedges nanopores codec. And first, how do we encode our data, like the text and the image files shown on the slides, into the physical DNA? Let's start with our session sequence designs. In these figures, we could find that we used to encode the image on like the SME logos. We used just 155 strands and 7 strands for the text, which is an introduction of our colleagues. And you may find that compared to the conventional 4-word configs, like the image and the text below, we use 8-word configs, which besides the ATCG4 bases, we also use 4 bases like the MKRY. And as you see in these tables, the M means half A, half C, and the K means half G, half T, and etc. And this This is the keys that we can double the informational density of our codecs. And on the right-hand side, you can find that the designs of our sequence. We first put the binary data into 144 nucleotide of the informational strands, and then we add 3 sections of 5 nucleotide of anchors. This anchors is served as the reference for the further consensus sequence alignment and the error corrections. And finally, we add 23 nucleotide of the primers and the 34 nucleotide of the barcode. And the barcode is mentioned in the last slide, is the sus tag, and mentioned in my colleagues, Mr. Junyao Li's presentations. And also we find that as we can't just directly synthesize the degenerative bases like MKRY. So when it's come to the synthesizes, we just use the half, like for example, if we want to synthesize YM basis, we just synthesize it by half of the batches use the A and half of the batches use the C, and other KRY is doing as that, etc. And let's come to our encoder and the decoder designs of the CHNs, which is mainly inspired by the headist, but we employ the degenerative bases to enrich their informational density. Let's start with the encoders on the left, and CHN use a hash-based approach to convert the binary data on the first rows into a DNA sequencing. As it encodes, it dynamically applies 3 constraints to the available alphabet. We'll consider 3 aspects. The first is the current 2 bits, and the last 54 bits using as hashing, and the current index. And the encoder also will check the length of the homopolars and the GC content to ensure the final sequencing we got is synthesizable, because we don't want the sequence cannot be synthesized, whatever. And when it's come to the decoders, we find that basically it is a brute force, Basically, it is a brute force ways to decode that. As you can see on these images, the decoders just we just use the brute force to construct all the tree structures that all possible solutions of the all possible binary data of the all possible sequence encoded by the corresponding binary data, and we just compared this possible sequencing with the sequencing we sequenced from the true nanopore sequencing, and we will calculate the added distance between the possible sequence and the sequenced sequence. And when we find that the sequence, the added distance is short enough, we just consider this sequence is the right sequence, and we will prune the other wrong one. However, as the trees may have a lot of computational resources, so we just limit the depths of the trees to about 50 or 100 layers. Yes. And here is a short image about the informational density and error tolerance rate of the CHNs, and you can find that with the degenerative basis, the 8-word config CHNs may have as much as twice of the informational density compared to the 4-word CHN, about 117 bits per basis, and in their resistance up to 15%. So let's come to the decoding pipelines of the CHNs, which can be done about less than 5 hours. And after sequencing, we must mention that we will first go to a clustering process that we use the anchors mentioned before and the barcodes that cluster the same strands of the sequence together and get a consensus read. We finally use this consensus read to send it into the CHN decoders to decode back to the tables. And here comes our performance. You can find that in the 20 minutes, we can recover the whole text, and in the 120 minutes, we can recover the whole image. And these two charts also illustrate this. And we only need to recover the whole text in about 60-fold coverage and the whole image in about 64-fold coverage. And here come up to my second part, the DPSME, which is a framework that uses the deep learning towards the single molecule encryptions. And here is also the old ones, the framework of the DNA storage. But if we find that, as I mentioned before, the modifications could alter the current and disrupt the base callers that could let the conventional base callers fail. So we just add the DNA modifications in the DNA synthesis process, and we also paired base callers is specifically for modification bases. So with these two steps, we got biological standardographic framework, and I can use these figures to explain it more clearly. And we use computational models on the encryption, as the Alice is the senders. She encrypts the data by synthesizing the DNA with some non-canonical bases, such as the modification. In our article, it is the 5-hydromethylation. And Eve is the evidence droppers or the unauthorized persons. He will using a standard base callers like the Bonito Guppy or some standard config. He will see only some garbled data or get a completely readout failure. And Bob is the authorized receivers. He will need the molecular case. In this scenario, it is the corresponding base callers to decipher his image or message. So how to train this, build these keys? The central challenge is training a base callers for these new modifications when no reference genomics or K-MER models will exist. And we solve this by a 3-step training pipelines list here. On the stage 1, we bootstrap the entire process by carefully design some 6-mer QC sequence that cover the whole 6-mer spaces. So the base callers may get initially recognitions of this nanopore current. And but as you see, after the first step training, the precision and the recall of the base caller is relatively quite low, nearly 0%. And at the second stage, we just use the CTC layers inside the base callers to get the data about the alignment data and use this alignment data to cut the current and get the K-MER tables. And then we can use these K-MER tables to simulate some great scale data sets, and we got the enhanced DPSMEs. And you can see that the enhanced DPSMEs got much more higher precisions, but still got a low recall rate, because the enhanced DPSMs only learns data from the simulations, not the real world. And on the third steps, we've just used the real world genomic DNA data, and we use the PCRs to replace the all Cs to 5-hydromethylation cytosines to do the modifications. Then we sequence this modified DNA sequence. And then we use these data sets to train the reinforced DPSMEs. As you see here, the reinforced DPSMEs got a jump on both the precisions and the recalls. And it is as well as the Bonito or Guppy base callers on the unmodified sequencing as it base call on the 5-hydromethylate modified sequence. So here's come to the real-world experiment of using the DPSME to deciphering or on concealment data stored in the DNA storage data set. And we found that when we want as you see, this experiment use our composite hydrogenation codec and the forwards codecs. So as you see that all text can be recovered at as low as a 60-fold coverage, And you can also find that in this diagram that up to 97% of the text and image can be recovered at the 60-fold, and up to 98% of image and the text can be recovered at 256-fold. And also you can see on the right figures that some unauthorized persons like the Eve with the standard base callers can hardly recover any meaningful data. They can only recover less than 5.4 tags at the 60-fold coverage. Also, this framework may have some potentials to a pathway to the fully encryption systems, because as you see that the researchers shows that there may be more than 429 modified bases has been discovered, and when we selecting 25 types to these modifications from the databases, and we just calculate the possible key length, and it can reach about 129 bits of key length, which is more than 128 bits, which is the recommendations of the NISTs. Yes. And here's come to our conclusions, and we proposed two computational frameworks. And the SAGE-N enables fast, robust, and high-density readout of the standard DNAs and solves the nanopore-specific Indel error problems. And the DPSME contributes to create a framework for the private and secure communications, use the modified DNA and its corresponding modified base callers, and solve the readout and the key management problems. Also, we have some future directions. As I said, the DPSME frameworks has not related to existed K-mer tables and the model weights. So we just want to show it can be transferred to some different nanopore flow cells like the ONT-R10 flow cells or other vendors like the cyclones. And also we may want to test the informational concealment with more alphabet, like this PowerPoint says. We may train some base callers with more than 4 letters, maybe 8 letters, 24 letters, etc. And here is the reference of my presentations. And thanks for the authors on these 2 articles, and thanks for the funding institutions that fund us research. And here's my end of the presentation, and thank you for listening.
Thank you.
Okay.
That was a lot of data. Yeah, you show a lot of things. Question, guys. Yeah.
Hi, thank you so much for your presentation. Cool data. I wonder, on your methylation experiments, how did you select the regions of DNA that you were marking, and how long were your fragments?
Maybe do you mean that how do I select the region of the DNA?
Yeah.
On the composite hedges, we just sorry, the computer is a little bit slow. On the composite hedges, we just when we want to set a specific strand of the DNA, we just use this, the strand ID, which is the SAS tag mentioned by our colleagues, and the anchors, these 3 sections, 5 nucleotide anchors to locate which strand of the DNA we want to extract. And the fragments, oh, sorry about that, I haven't put the data on the slides. Maybe you can find it in my supplementary information of my composite hedges articles. The articles may demonstrate that. Okay.
Thanks.
Thank you.
Any other comment?
You showed the potential for using many different letters with some modifications.
Yes.
But I guess there is a limit to this. Maybe all these modifications are not detectable by Nanopore, Did you make some tests with other modifications than methylation, for example?
Yes, we have in our labs we have a test on some common modifications like the 5MC and 6MA. But as this researcher says, the 425 modifications, maybe many of them may be still unstable or hard to get. So in the future, maybe we can try some others, or we also propose that we can use we can split the informations into many parts and encryption it part by part with the different keys. That may also contribute our key length, and may also get up to the recommended key length by the IST. Yes.
So my question was not on the stability of this, but I guess many of these modifications will induce a current change that cannot be distinguished on a nanopore.
Oh, you mean this? And your questions maybe make sense. But we just test the 5MC and the 6MA. These two modifications, the current of these two modifications are easy for us to distinct, and these two modifications, the current caused by these two modifications, can easily disrupt the current base callers and also make them unable to read some meaningful data. But for the other modifications, we may need to do further investigations, I think.
I have another question.
Mm-hmm.
Oxford Nanopore developed some dedicated base caller for detecting methylated bases. Did you compare with your base caller?
Yes, we compare that in these figures, because as I think that the nanopore. First, I must mention that as we use the R9 flow cells, the ONTs only provide some R9 flow cell detection method on some motifs, not the whole regions. And secondly, we found that most of the ONT-based callers are trained with some sprays modifications instead of our heavy or the intensity modifications, so they do not perform well on this data set. But however, if the ONT guys want to train some base callers like us, they may still get some meaningful data readout. Yes, I see.
I have a question. It was on this slide I had a question, the one you were. You said that, so if the person that is trying to catch the message cannot decode it at 16 times coverage, but if you increase the coverage, can you then... there.
Yes. And I think in these figures, I use the 16-fold coverage because as we calculate the coverage, we just use the SUS tags or the barcode regions to classify whether it is a part of the strand or it is not. But however, as we sequence about 1.9 million reads from the nanopore sequencing, and we can hardly find any strands more than 60-fold coverage, so we just use the 60-fold here.
Okay, so the coverage is not...
yes.
Okay, thank you very much.
I think we have one more question here, right?
This was more of a comment on the nanopore modification detection capabilities. So with R10, we now have 4 DNA modifications that can be detected, and 8 RNA modifications as well. So even if all of them will not induce a change in current, most of them can and will be able to train models for them. So there's a lot of capabilities there.
Okay, I see. So I also see the ONT, the R10 specific models released by the ONT, and it can recognize the modifications without of the motifs. So that may be a challenge for our current framework. But I think with some, if we could find some modifications that is rare enough or which could alter the signal enough, there could be still have some feasibilities for this framework, I think. And we will try this on the R10 chips later. Yes. Okay.
Continue the discussion after the coffee, right? Thank you again for the very interesting presentation. A lot of work here. Thanks.
Okay, thank you for the listening.